Hi, welcome all to the lecture on diagnosis of infectious diseases. Okay, so end of the lecture, you'll know various methods in diagnosing infectious diseases, as well as you'll have an understanding on a specific technique used in isolating and identifying pathogens from clinical samples. Okay, why we need these diagnostic tools? Okay, diagnostic tools are very important in order to identify the infection so that a proper treatment can be initiated and also to improve the clinical outcome as well as improve the surveillances. And this means especially when it comes to the resistant cases. So from these diagnostic tools, you can know whether a particular bacteria is resistant to particular antibiotic or not. And from there, you can initiate a proper treatment. Okay, an infection can be diagnosed using various methods, so such as differential blood counts, hypersensitivity reactions, antibody titers, and also isolation and identification of pathogens, which can be divided into collection of specimen in isolation and identification of pathogens. So in this lecture, we'll go through one by one. Okay, the first one, differential blood counts. So as we know, blood has two components, which are plasma membrane, which is made up of 55% of the blood, and another 45% is the cellular elements. Plasma membrane is consisting water, ions, plasma proteins, and other substances in the blood. On the other hand, cellular element is containing red blood cells, which is erythrocytes, and white blood cell, which is leukocytes and platelets. Okay, differential blood count is used to determine the relative number of white blood cells as a diagnostic indication of an infection, and it also provides a general indication of the nature of the infecting agent. For example, systemic bacterial infection and some localized infection will lead to leukocytosis. Leukocytosis is a condition where there is a high number of leukocytes, or in particularly neutrophils. For example, if someone have a Staphylococcus septicemia, so there will be like a higher number of neutrophil compared to the normal. And on the other hand, typhoid fever, brucellosis, and viral infection will lead to lower number of neutrophil count. Okay, this is a sample result from a differential blood count. So you can tell that is a white blood cell, red blood cell, hemoglobin, and so on. So from these readings, so you can analyze whether a particular person has an infection or not. And this is actually the normal range. Okay, the second one is the hypersensitivity reaction, or also known as a skin testing. Hypersensitivity reaction is an abnormal condition characterized by an exaggerated response of the immune system to an antigen, and is often used for screening method and cannot be used for positive diagnosis of a disease. So which means after you know it's positive in a hypersensitivity reaction, you have to go through another testing to confirm the disease. Okay, this picture is showing the sample after the hypersensitivity testing. So usually you can see a red zone as shown here. So when you go to the hospital, you will be injected with the antigen that derived from the test organism. Usually the injection will perform intradermally, which means under the skin. And after the incubation period, you will get a red zone. Okay, usually the incubation period is 24 to 72 hours. And then these red zones will indicate a particular person is exposed or become sensitized to the specific antigen. And this test is always performed for the tuberculin reaction to diagnose a tuberculosis. Okay, the following one is the antibody titers. So antibody titer is a lab test that measures the presence and amount of antibody in the blood. And it can be measured as an indicator of a disease and usually rise in the antibody like a fourfold increase during the active phase of an infection. And antibody titers can also be used when the pathogen cannot be isolated, so which is case for a viral infections. And usually the results expected as the greater dilution titer that still gives a positive results through a hemagglutination. Okay, I will just briefly go through on this antibody titers. Okay, what you usually do, so you need to dilute the blood and then also you add with a similar amount of antigen, and then you also should have one control. And usually when the antigen reacts with the blood, and then it will form a antigen antibody complex at the bottom of the well. Okay, in this case all these four as form the antigen antibody complex met, except for starting from this one. So therefore This concentration 1:160 considered as a positive reaction because it produced the MAD of antigen antibody complex. Actually, this topic is not really into your syllabus, so as for now, just briefly knowing this is sufficient. Okay, the last method of diagnosis is the isolation and identification of pathogen. A definitive diagnostic of an infectious disease involves the collection of specimen, isolation, and identification of antigen specifically associated with the microbial pathogen. So therefore, in this lecture, in the last bit of the lecture, we'll go how a sample collection and isolation can be performed, and also we'll briefly go through on the identification of pathogens. Okay, in this lecture, we will focus how the sampling can be done in these areas. Okay, let's start with the respiratory tract. So respiratory tract can be divided into upper respiratory and lower respiratory. So upper respiratory is from your nasal cavity up to larynx. If it's a lower respiratory, from your trachea up to the lungs. Okay, let's begin with the sampling in the upper respiratory tract. So if it's a bacterial infection, the culture will be collected using sterile cotton swab, looks something like this. And then this cotton swab will be placed in a sterile transport media and then stick onto agar plates. And the agar plates will be incubated at 5 to 10% of carbon dioxide. And these are the common pathogens and the media that can be used. For example, Streptococcus pyogenes and Streptococcus aureus, we use blood agar. If it's a Haemophilus influenza and Neisseria meningitis, we use chocolate agar, and so on. Okay, on the other hand, if it's a viral infection, the growth of the virus will need tissue cultures. So what we usually do, so the cotton swab, which is already swabbed on the upper respiratory tract, will be transferred into the cell culture or tissue culture. And then we will look for cytopathic effects. And some people also will do serological methods, which is based on antigen-antibody reaction, as we learn in the antibody titers. And some rich laboratory will also do electron microscopy, which is to identify the viral isolates under the microscope. Okay, the sampling of upper respiratory tract is known as a nasopharyngeal swab procedure. There's a video here on the nasopharyngeal swab.
Check for nasal obstructions. Open swab package. Gently insert the swab into the nostril. Keep the swab near the septum floor of the nose while gently pushing the swab into the post-nasopharynx. As a visual reference, the swab should be inserted about half the distance from the opening of the patient's nostril and the ear. Rotate the swab several times. Gently remove the swab and perform the test according to the manufacturer's instructions. If the specimen testing is not performed at the point of care, refer to the manufacturer's instructions for transport and storage recommendations.
Okay, next is the lower respiratory tract. So for the lower respiratory tract infections, sputum will be frequently collected. So sputum is a type of a thick mucus that is produced by the lung when they are diseased or damaged. Usually it is a mixture of saliva and mucus coughed up from the respiratory tract, and it will look something like here. Okay, and then the common pathogens that can be found in the lower respiratory tract are Streptococcus pneumonia, Streptococcus aureus, Streptococcus pyogenes, Klebsiella pneumonia, Haemophilus influenza, and usually these bacteria need to be plated on the appropriate media. And apart from them, for TB and fungal infection, usually they will be differentiated using a staining method. For example, for tuberculosis, differential staining such as acid-fast staining will be performed. Okay, the following one is the cerebrospinal fluid or CSF cultures. And usually if it's a patient is suspected with the infection in the central nervous system, then you have to go through a CSF. And usually CSF is performed by a lumbar puncture. And for bacterial infections, usually people will do gram stain and culturing using a suitable media. And the common media usually are blood and chocolate agar. And this is the example of pathogens that can be found in the CSF cultures. Okay, this video will show us how CSF is performed. So let's watch it together.
use a little lidocaine. It's the numbing medicine, okay? You're going to feel a stick and a burn, okay? Just try and sit as still as you can, okay? This is the time when you really need to arch your back out as much as you can, okay?
Yeah, it is a very interesting music for this video. Okay, the following one is the blood cultures. So blood culture is pretty straightforward. So the blood is withdrawn and then grown in the suitable media. And then the commonly used media are blood agar, chocolate agar, McConkey agar. And usually one of the things people do for blood cultures are they incubate under aerobic and anaerobic conditions. Okay, urine culture. So in order to do the urine culture, so we need a midstream urine sample to minimize contamination with the normal flora of genitourinary tract, so particularly in females. And a high number of microorganisms, for example, more than 10 to the power of 5 bacteria per mil is an indication of an infection. And the commonly used medium are blood agar, cysteine lactose electrolyte deficient agar, McConkey agar, and eosin methylene blue agar. And these are the common pathogens that can be found in the urine samples. Okay, next is the urethral and vaginal exudate cultures. Okay, urethral and vaginal exudate cultures are used for the detection of microorganisms that cause sexually transmitted diseases. And common pathogens are Neisseria gonorrhea, Chlamydia species, and Treponema pallidum. And other pathogens including Haemophilus ducreae, Streptococcus pyogenes, Staphylococcus aureus, and Candida albicans. And the common mediums are Thaye Martin for Neisseria gonorrhea, blood agar, chocolate agar, McConkey agar, and Sabro agar for Candida albicans. And on the other hand, for the Treponema pallidum, the detection usually performed using a dark field microscopy, and then sometimes also use fluorescent antibody staining. And people hardly use fluorescent antibody staining because it's quite expensive. Okay, next is the fecal cultures. So stool specimens are required for the fecal cultures, and usually fecal cultures are used for the diagnosis of intestinal tract infections. And the common pathogens are Salmonella, Shigella, Enteropathogenic E. coli, and so on. And the medium are used often selective and differential media. For example, xylose lysine deoxycholate agar. So this media can differentiate between Salmonella and Shigella. So please look at this figure. So the xylose lysine deoxycholate media usually red in color. So when Salmonella grows in the plate, which is figure C, and then this Salmonella can ferment the xylose to produce acid, and this will change the media into yellow in color. So if you see the media become yellow, that means there is a Salmonella. And on the other hand, if the media remain red, so that shows the presence of Shigella, because Shigella cannot produce acid, and then therefore it remain red. On the other hand, for the viral infection, we can inoculate into tissue cultures, and then we see the cytopathic effect, as I mentioned earlier. And then some people also do serological procedures. Usually it comes with the diagnostic kits, and usually it's quite expensive. And then there are also electron microscopy techniques where you can also see viral infections. Okay, next is the eye and ear cultures. The commonly used media for eye and ear cultures are blur agar, chocolate agar, and McConkey agar. And one of the trouble or issues with ear and eye cultures are to differentiate between bacteria and viral. And then one of the things people usually do is they perform gram staining to identify the bacteria. So once they identify whether it's a bacteria or viral infection, and then a proper treatment will be initiated. Okay, next is skin lesion cultures. So skin lesion cultures are usually performed to detect bacterial and fungal infections. And one of the ways to identify them is via direct microscopic examination of the skin. And this is very useful for the fungal infections. And recently, we also have a skin biopsy. So skin biopsy also can be performed where a portion of the skin is removed and then cultures in the particular media. And then we do the identification. Okay, I have a video here to show how the
Skin biopsy
is
procedures.
So let's
Your
watch it
doctor will
together.
inject your skin with a local anesthesia to numb the area. Shave biopsies are usually performed on surface lesions. During this procedure, your doctor will use a sharp scalpel or razor blade to thinly slice or shave the top layer of the skin lesion. This type of biopsy does not require stitches. Doctors usually perform a punch biopsy for deeper skin lesions. Your doctor will rotate a circular hollow blade around the lesion until it cuts completely through the epidermis and dermis. He or she will remove a small cylinder of skin, usually smaller than the size of a pencil eraser, containing all layers of the skin and lesion. Depending on the size of the sample, the area may or may not be closed with stitches. An excisional biopsy is larger and deeper than a shave or punch biopsy. It is used for conditions such as cancer, which must be entirely removed. Your doctor will go as deep as is necessary to remove all of the area visibly affected, as well as some unaffected tissue around the outside of the lesion. The incision is then closed with stitches. After the biopsy, you will need to keep the area dry and cover it with a sterile bandage or gauze dressing for 1 to 2 days. Antibiotic ointment should be applied until it is completely healed. Acetaminophen or ibuprofen can be taken for any discomfort at the biopsy site.
Okay, so we have done with how the sample collection and isolation can be performed. So let's move to the next topic, which is identification of pathogens. So the method chosen must be accurate, reliable, and quick for an accurate identification of pathogens. So generally there are 3 methods. One is metabolic, second is a serological, and the third one is the gene probe. Okay, the first one, the metabolic identification. So metabolic identification is used to differentiate the bacteria by the substrate they utilize and the product of the metabolism. So now we have a kit in the market which is known as a Analytical Profile Index API 20E system. And this is a system that is able to classify the bacteria based on their biochemical tests. And this allows a fast identification of the pathogenic bacteria. And usually for the anaerobe, we doesn't use the API 20E system. We use a gas liquid chromatography or GLC for detection of fatty acids. So each anaerobic bacteria will have like a different fatty acid content. So based on that composition, we can able to identify the pathogenic bacteria. And these are some of the volatile fatty acids. Okay, this is the API 20E strip for the Aeromonas hydrophila. So Aeromonas hydrophila can cause a broad range of infections, which is including septicemia and meningitis. So this shows the strip of consisting of wells containing dehydrated substrate to detect enzymatic activity usually related to fermentation of carbohydrate or catabolism of protein or amino acids. So perhaps you can read on your own. So if you have any question, please post in the question box. Okay, next is serological identification, and this is useful in identifying pathogens that are difficult or impossible to isolate on conventional media or not easily distinguished by biochemical testing. And this test is based on the blood test that look for antibodies in the blood and focuses on proteins that made up the immune system. So I will upload the video in the e-learn, so perhaps you can watch the e-learn to know more about the serological identification. Okay, the last method is the gene probe identification. So in this method, gene probe will be used. Gene probe is a small molecule of single-stranded RNA or DNA with a known sequence of nucleotide that is used to detect or identify a homologous complementary nucleotide sequence in the sample. So usually for this method, you do not need to culture the pathogen. So which means you do not need to do a isolation. So you can directly use the clinical sample. And one of the advantages of gene probe identification is that it is very speed, is a rapid method and accurate. Okay, that's all in this lecture. Thank you for your attention. And then I will post some extra videos in the e-learning so you can watch the video and then get some extra information from there. And then don't forget, if you have any question, please post in the e-learning as well. Thank you.